Hi, my name is Taylor, and I study how the brain changes throughout the day. Our body has an internal clock that regulates our functions and repeats its cycle approximately every 24 hours. This cycle is called a circadian rhythm. One of the first examples that I think of when I think of circadian rhythms is the sleep cycle. Around the same time every night, you probably begin to feel tired. And if you didn't have to set an alarm, you would wake up around the same time each morning. But sleep isn't the only process that repeats itself each day. Your daily trends in heart rate, digestion, and even pain tolerance are guided by circadian rhythms. In my research, I look at how specific brain cells called astrocytes change their activity throughout a 24-hour day in rats. My project focuses on the hippocampus, which is the part of the brain that is involved in helping you learn and store memories. This learning is achieved by neurons, which are a special type of nerve cell in the brain that work together to transmit electrical impulses throughout the nervous system and within the brain. But these neurons don't act alone. There are many other types of cells in the brain that help support this communication. Astrocytes are found to be very close to neurons, and they help to keep neurons healthy, and they can even influence the strength and rate of electrical transmission in the cells. Our previous research in my lab has found that astrocytes themselves also show 24-hour activity. My goal is to further characterize how astrocytes differ between day and night. Using a fluorescent protein marker, I can take images of brain slices under the microscope, and only the specific protein that I want to see will fluoresce in the final image, and I can compare them between time points. The specific protein that I'm interested in is called phosphoesrin, which is a marker of movement and activity in astrocytes. Astrocytes are star-shaped cells that have many fine processes that extend out towards neurons or retract back into the body of the cell. By imaging these brain slices at different times of the day in the 24-hour cycle, I hope to determine when astrocytes are most active and moving and when they are closest in proximity and most connected to neurons. After determining this, I plan to add a chemical to the brain slices that disturbs astrocyte function by not allowing them to communicate with other cells. By seeing how this affects the 24-hour activity cycle, we can see how unhealthy astrocytes negatively impact the brain. By learning more about how astrocytes and neurons interact throughout the 24-hour cycle, I hope to gain a greater insight into how our system of learning and memory works throughout the day. Thank you.